afternoon, everyone. We are Group 10, and our team members are Infinity Woodruff, Nathaniel Goff, Harrison Stuckey, and myself, Daisy Blasi. Today we will be presenting our research on the effects of pollution on land and aquatic plant species. Our study is going to examine how pollution impacts plants at both the phenotypic and genotypic level, with the primary focus on terrestrial and aquatic ecosystems. By understanding how plants respond to pollution, we can contribute to conservation efforts and predict long-term environmental impacts. Before we get started, here is a quick overview of what we'll be covering today. We'll start with background information, outline why this research is important, then we'll present our research question and methodology, detailing our approach to the data collection and analysis. Next, we'll discuss our timeline and expected outcomes. Finally, we'll wrap everything up with the significance of our research, followed by our conclusion and references. Pollution is a major environmental stressor affecting plant ecosystems. Industrial activities, urbanization, and agriculture release harmful pollutants such as heavy metals, phosphates, and volatile organic compounds. These contaminants impact plant biology in two main ways. First, phenotypically, by altering physical traits such as stomatal density, root structure, and pigment composition. Secondly, genotypically, by triggering differential gene expression and mutations that influence plant adaptations. Our research aims to explore how pollution impacts plants differently in both terrestrial and aquatic environments, while addressing key gaps in current scientific understanding. To dive a little bit deeper into our background, plants respond to pollution in distinct ways depending on their environments. Terrestrial plants are exposed directly to airborne pollutants and contaminated soils, leading to stress-induced changes in their morphology and gene expression. Whereas aquatic plants are going to absorb pollutants from the water directly, which has been linked to biodiversity loss, particularly in species sensitive to phosphate accumulation. A key gap in existing research is the comparative analysis of pollution resistance across different plant species. Some invasive species may be more tolerant to pollution, while native species are going to be much more vulnerable. By investigating these differences, we're hoping to provide insights that support conservation efforts, phytophthora remediation strategies, and agricultural sustainability.
Now that we've finished discussing the background of this project, we're going to go into what we're actually questioning. So we want to know, how has pollution on land and in aquatic environments affected plants both phenotypically and genotypically? When we're talking about phenotypically, we're talking about observable traits. And when we're talking about genotypically, we're talking about genotypes, something such as gene expression and mutations across the terrestrial and aquatic environments. If we understand these responses to pollution, this can help inform conservation strategies and predict ecosystem resilience. Some of the possible plant phenotypes that we expect to see are stromatal density, root structure, and pigment composition having some sort of altered change. As far as gene expression goes and mutations, there are a lot of gene expressions that we expect to be changed throughout pollution's impact. And some of these are such as polluted stress response genes, having stress response genes that are been impacted by pollution can possibly reduce their effectiveness. Specifically, genes involved in detoxification, oxidative stress management. Some of these could be elevated, some of them could be reduced. It just depends on the situation. So we're going to be looking into this.
Hi, my name is Infinity, and I will be beginning the discussion on the methodology for Group 10's course project presentation. So ideally, we would want to start off with some known samples that we collected, do extraction, library prep, sequencing, and then do analysis. But within this capstone course, we will be starting with some open access data, which we will collect, and then we will start doing analyses from there, meaning that we're going to be skipping basically steps 1 through 3, and our project will focus on the analysis, step 4, to answer our course project question. So I will be responsible for the NGS analysis part of our course project. I will be doing foundational NGS analysis using Bioconductor in R. Bioconductor is an open access repository within R that has tons of different packages that would be great for analyzing NGS data. And whenever I say foundational NGS analysis, I'm referring to things like what biological strings are we using? How big are they? How many base pairs? Maybe things like where are certain genes located? Also, maybe I'll look at the alignment of our data to a reference genome. Maybe we can take some sequence data that has already a read count to it, and we can determine, how reliable is the data. So all of these different analyses, which I would consider foundational, are not typically very complex analyses, but they do set the foundation for our course project and allow us to have a better understanding of our data. That way we can move forward into more complex, things like statistical analyses. So in order for me to be able to perform this NGS analysis, I'm going to be acquiring the skills through DataCamp. There is a skills track that they offer called Analyzing Genomic Data in R. So I will be completing that to perfect my skills, and then also I'll be going to YouTube where there are tons of people who post videos of them actually using the packages with different types of data. So this combination of using the skills track within DataCamp and also following YouTube tutorials will allow me to be able to practice and perfect my skills for this project. Some of the main packages that I will be using within Bioconductor are biostrings. This is good to do manipulation of large biological strings, and also genomic ranges. This is good for finding target regions of the genome. Both biostrings and genomic ranges would be good for annotation and aggregation of our data. I will also be using EdgeR and DSeq2. These packages are really good for identifying genes and pathways, along with determining read counts. These two packages are good for, proving our confidence in the variability between different plant species. I will also be using Lemma. This is good for fitting expression data to a linear model, which would then be able to tell us what are the effects of polluted environments on different plant species. Lastly, I will be focusing in on RBowtie2 and GViz. These two packages are good for aligning data to a reference genome and then plotting that data. And this would be really good in the identification of mutations within different plant species that are inhabiting polluted environments.
During our project, I'll be taking on the field data collection and morphological assessments. We'll be using this to analyze how pollution can affect plants, and we'll need to collect comprehensive environmental and biological data. At our site selection and categorization, we're going to be looking at plant samples from industrial, agricultural, and urban sites with varying pollution levels. We'll also be looking at soil and water samples that will be analyzed for pH, heavy metals, and nutrient levels to measure pollution's intensity. From there, we're going to be looking at our plant sampling and trait analysis, where we're going to be measuring stomatal density, root structure, and pigment composition to assess observable adaptations to pollution. And the key focus will be comparing native and invasive species to determine which plants will show a greater resilience. Lastly, we'll be looking at our molecular analysis. RNA will be extracted from plant tissues to examine gene expression changes due to the pollution exposure. Quality control measures like a bioanalyzer and fluorometry will ensure high-quality genetic data. By integrating field and molecular data, we'll be able to correlate environmental pollution levels with plant stress responses, providing valuable insights into adaptation mechanisms. I will also be focusing on data manipulation and pre-processing within Python. Once our data is collected, we will need to process, clean, and analyze it efficiently, and that's where Python is going to play a crucial role. First, we'll do pre-processing environmental and sequencing data. I will use pandas and NumPy to structure data sets, handle missing values, and normalize pollutant concentrations across sites. Next, I'll be looking into genomic data processing, where BioPython will be used to filter low-quality reads, trim sequences, and prepare data for differential gene expression analysis. And then lastly, I'll be looking into data visualization and interpretation. Using SciPy and Matplotlib, we will generate statistical distributions and visualizations of pollutant gradients. This will allow us to correlate pollution levels with plant stress responses, reinforcing the connection between environmental stressors and plant adaptation. By using Python, we will ensure efficient data handling, enabling us to extract meaningful insights that contribute to our overall research objectives.
Another method for exploring the research of pollution and its effects on plants, both land and aquatic, is using linear regression. By using linear regression, it can help quantify these relationships between pollution factors and plant traits. Pollution factors are the independent variable. So something such as lead would be lead within the soil levels. That would be the independent variable. And what's being the dependent variable is the plant trait. Something such as like gene expression levels or whatever it may be. The model down here is just showing an example of how it's used in R. And essentially, this linear regression will help us identify whether pollution significantly affects the traits or if it does not. These skills can be acquired through using data camp and this statistical fundamentals in R, as well as looking at some old notes from the previous classes in LSC 540 and LSC 541. This presentation had not been updated, but it has been edited to say LSC 541.
Hello, my name is Harrison Seki, and I'll be going over the last of the methodology section and the timeline for our project. In addition to the NGS analysis and linear regression, we will also be applying statistical genetic methods in the form of genome-wide association studies, or GWAS, to identify genetic variants associated with pollution tolerance. The GWAS methodology can be broken down into 4 parts. First, we will obtain genotypic data in the form of SNP data from plant species exposed to varying levels of pollution. This data will be sourced from public repositories or generated from our next generation sequencing dataset. In addition, phenotypic traits such as growth rate, leaf area, root structure, etc. will be measured or extracted from databases like the TriPlant Trait database. Once the datasets have been created, we will filter and normalize the data in applications such as R or Python and export them to be used in specialized GWAS software called Tassel. And with Tassel, we will perform association analysis to identify genetic markers significantly correlated with phenotypic traits under pollution stress. The software employs statistical models to account for population structure and relatedness, reducing false positivity and ensuring robust results, which I will go over briefly in the next slide. Lastly, we can export the results and data to be visualized and compare with our gene expression analysis. Ultimately, GWAS complements linear regression and NGS analysis in that we can achieve a more holistic understanding of how pollution affects plants. Linear regression provides a high-level view of the environmental impact, while GWAS delves into the genetic mechanisms driving these changes. Together, these methods enable us to identify key genetic markers for pollution tolerance, predict how different plant species or populations will respond to pollution, and develop targeted strategies for breeding pollution-resistant crops or conserving vulnerable species. So, as briefly mentioned in the previous slide, the GWAS software that will be used is called Tassel, which stands for Trait Analysis by Association, Evolution, and Linkage. We chose Tassel in that it has a user-friendly interface, scalability for large datasets, provides comprehensive analysis, and can easily be integrated with other tools such as R or Python for our linear regression and data preprocessing. Tassel accepts various file formats, including VCF, variant call format for SNP data, and HapMap for genotype data. And for our phenotypic data, we can import them into Tassel easily in tabular format, where each row represents a plant sample and each column represents a specific trait. The core functionality of Tassel will be the use of its statistical models for GWAS. TASL supports multiple statistical models, including general linear models, or GLM, which is a basic model for testing associations between SNPs and traits, and mixed linear models, or MLM, which is a more advanced model that accounts for population structure and relatedness, reducing false positives ultimately. Additionally, TASL calculates p-values for each SNP trait association and applies significance thresholds, such as Bonferroni correction, to identify statistically significant loci. Lastly, once significant SNPs are identified, TASL can be used to annotate these loci with functional information, such as gene names, pathways, and biological roles, and we can then export our findings for further visualization or analysis. And to the right here is an example of what the software looks like, and in this image displays a linkage disequilibrium plot. We plan on acquiring these skills by practicing with the TASL software and watching any tutorials and reading textbooks alongside with completing statistics modules and DataCamp. Next, I'll briefly go over our timeline for our project. We currently expect this project to be completed in about 12 months time, where each course section takes about 2 months. First will be literature review and data collection, where we will review existing studies and gather data from various databases. Next, in months 3 and 4, after we identify our data source, we will begin to clean and pre-process our data sets for our experiment. In months 5 and 6, we will perform our NGS analysis by performing differential expression analysis in Bioconductor. This will be followed by a phenotypic trait analysis to correlate phenotypic traits with our NGS analysis in months 7 and 8. In months 9 and 10, GWAS will be conducted to complement our other analyses. And lastly, we will integrate datasets and create visualizations of our results to finalize our study. And that concludes our timeline and work plan.
So why is this project that we are going to be completing significant in the first place? There are many different types of pollution. Some of the major ones are land, water, noise, and air pollution. Of course, within this project, we will be focusing on land and water pollution. Now, some of the main things that are considered, really detrimental pollutants are plastic, of course, and also the release of hazardous compounds, for example, phosphate. Now, these different pollutants are, being released into different environments, land and aquatic, and it's breaking down very fragile ecosystems. So, there are conservation efforts that are out there, trying to combat this, but it is very apparent that more is needing to be done. Furthermore, on significance, pollution on land and in aquatic environments very clearly outpaces current conservation efforts. So even though there are efforts in progress right now to halt or stop pollution that is happening, it is very apparent that more is needing to be done because pollution is outweighing efforts that are happening tremendously. Some of the major contributors are urbanization, mining, industry, and agriculture, which when you think about it, these 4 major contributors make up the majority of, jobs out there. So it's definitely a huge issue. From these contributors, there are hazardous compounds that are being released into ecosystems, damaging them, and even wiping out species, specifically plant species. So I think that this project would be very important for investigations into the toleration and the resistance to pollutants to protect ecosystem balances. So the 2 graphics that I have here, I think, are, very good depictions of pollution and how they are affecting plants. So the first graphic shows how things like air pollutants, which isn't a focus of this project, but it definitely goes hand in hand with the land and aquatic pollution. So things like effects on, tree productivity. So maybe there are fruit trees that are in environments that are being polluted, so they're not producing the fruit that they need. And then that in turn, is kind of like a domino effect. We won't, see those fruits within grocery stores, and then it becomes like a food source issue. Additionally, there are things like effects on plant height growth, plant leaves falling, and plant roots weakening. And when you think about it, all of these things combined make it really hard for these plants to survive. So they end up being wiped out. So one big thing to, battle this pollution issue is phytoremediation. So some of the major phytoremediation aspects are things like phytodegradation. And this is when pollutants are broken down from enzymes. There's also phytostabilization, which is when pollutants are immobilized within the soil. Also, phytoextraction is whenever pollutants are accumulated in harvestable parts of plants. And also phytostimulation is when pollutants are degraded by growing roots and microorganisms. So there are things that plants can do to be able to withstand and resist, tolerate these pollutants that, come into their environments. But it is very apparent that, more projects like this one that we are doing is needed in order to, better equip these plants, whether it's through things like gene editing and things like that, to amplify their toleration and resistance to pollutants. Because we don't realize it that these pollutants are really destroying environments, and it will in turn affect us. We don't realize it until, it's gotten to the point where it's just horrible, or, a plant species is extinct and things like that. The ecosystem is just breaking down. So I think that this project will be a good driving force for these investigations in order to protect our ecosystem balances in the future.
To conclude, as we've explained before, we expect that pollution heavily alters plant morphology, physiology, and genetic expression. There's a suspectability that invasive species are going to be more resilient to native species based on the hardiness and ability to reproduce and thrive within various environments. Regression models can help quantify the relationship between pollution and plant adaptation, as well as the other methods that were described. This matters because this supports conservation and land use policies, and if we understand this more, it can help us understand biomarkers for pollution and stress in plants and help provide ways and areas for us to improve on in order to help keep the sustainability of the environment.
I just want to go ahead and thank everyone one last time for taking the time to listen to our presentation on the study of aquatic and terrestrial plants within pollution and how it is all intertwined and affected. These last two slides are just going to go over all of our references that were used. Thank you again.